Welcome to Zooming into Cells.
My name is Madeline, and I'm a research scientist here at the University of Michigan in the Department of Biological Chemistry. Biological chemistry, two really big words. Pretty much when we put them together, we get biochemistry. What does that mean? Pretty much it means we study the chemical mechanisms that are responsible for keeping you alive. And these mechanisms occur in a cell.
How do we study cells?
All right, we are currently standing in a tissue culture room. A tissue culture room is a sterile environment where we can grow cells in a dish. I put on my gloves. These are required to not only protect me, but also the cells.
Now you can see that I am preparing my tissue culture hood. This requires that I bring all the different ingredients I need into the sterile environment to be able to prepare my cells for experimentation. You can see that I am gathering my cells from the incubator. The cells are in pink liquid, which you can see in the dishes. This pink liquid includes sugar to give the cells food to keep living, as well as other stuff that keeps the cells really happy, or what we like to call in a homeostatic state. Right now, you can see that I am taking clear liquid and I'm washing off the cells to get rid of any debris. I then will add one more liquid after I wash them off, and this liquid, which we call trypsin, that's going to come from the little bottle, is required to cleave the cells off the plate to be replated afterwards in a new setting. This is important because as cells grow, they need to have room, and so you constantly need to help them live in different size dishes. The trypsin that we put on the cells requires 5 minutes to activate. As you can see, after 5 minutes, I retrieved my dishes that I'm now looking at in a microscope. When we zoom in and we look at the microscope, we can see our cells in our dish. Isn't that so fascinating? So now you can see that I'm knocking the dish, and this is going to cause my cells to move around. We need the cells to move around on the dish and no longer be stuck to the bottom to be able to replate them, just like we discussed before. So as we zoom in, you're going to now see that the cells are moving around. By tapping the dish, I knock the cells from adhering to the bottom, and now they'll be able to be replated, put in a new dish, and be given more area to grow. We are now going to add the pink liquid that contains the sugar and other stuff that the cells need to stay alive. We're going to give the cells lots of this so that they have plenty of nutrients to stay healthy. Now we're going to replate a small portion of our cells into these small little circular dishes you can see on the left of me. These small little circular dishes are what we're going to use to do our experiment on our cells later. Now let's go back in the lab and let's talk about this experiment that we're going to do together.
To design our experiment, let's start off with going through the scientific method. The scientific method is made up of 4 different parts, and that first part is called the research question. To think about what we want our research question to be, let's go back over to the cell biology. One part of the cell that we're going to focus on is this compartment called endolysosomal system. And to remember, as a reminder from before, this compartment is responsible for recycling the machinery that the cell no longer needs. However, sometimes this recycling doesn't work properly and damage can occur to the endolysosomes. When this damage occurs, this can lead to a buildup of toxins. And when this happens to the cells that are in your brain called neurons, this can lead to diseases. And one example is Alzheimer's. Our research question is going to be, how are endolysosomes damaged? And to study this research question, we need two components. The first component is a dye. So what's going to happen is we're going to add a dye to our cells in our dish. This dye is green, and it's specifically going to be bright only in the endolysosomes. The second component we need to answer this research question is a drug. This drug is called LOME. The second part of the scientific method is the hypothesis. And this is our prediction of what's going to happen when we add this drug, our first component, to the cells that have the dye in them, our second component. So what's important to know before we make our hypothesis is what this drug does. This drug, LOME, when we add it, this drug causes damage to the endolysosomes. And so when we add this drug, we're predicting it's going to create holes in the endolysosomes. And when holes happen, this dye is going to leak. What's really cool about this dye, when it leaks, it's going to travel through the cell and end up in the nucleus. And what's really cool about this dye is that it's going to increase in brightness when it gets to the nucleus. So we're able to measure the leakage from this drug to the endolysosomes by the increase in fluorescence in the nucleus. All right, our hypothesis is, and reminder, this is our prediction of what's going to happen. Adding LOME, our drug, to cells causes damage, and that this damage then leads to the dye that we added to leak, that then causes brightness in the nucleus. The third part of our scientific method is the experiment. We're going to use the experiment to answer our research question and test our hypothesis. And the one key component of the experiment is a control. And so our control that we're going to use are cells that are not treated with the drug, and compare those to the cells that are treated with the drug. And this will allow us to understand, when we add that drug, is there a difference that happens in the dye leakage versus when we don't add the drug? The fourth part of the scientific method is results. And this is what we're going to find out when we do our experiment on the microscope. So let's go.
All right, you can see that I am placing our little dish that has our cells in it for our experiment on top of our large microscope.
All right, so as you saw, I brought the cells in from the tissue culture room, and we have them here in the incubator. This incubator is at the same temperature as your human body. So the cells are very happy, and you can see right here, here are our cells. And so what's important is this green dye we are talking about. And you can see that the nuclei has a starting brightness because some of that dye also is in the nucleus
Due to nonspecific loading.
So remember from before, when we were talking about the experiment part of the scientific method. In one of these compartments, we're going to add no drug, and in another, we're going to add a drug. And so therefore, we'll be able to see what difference happens when we add that drug. I'll go get on my gloves and let's add the drug.
We are going to begin by adding no drug. As you can see, I am taking substance from the tube that has no drug in it, and I'm going to add this to one of our chambers in our dish that contains our cells. Next, we are going to add our LLME drug. You can see that I'm going to take media from the tube that has LLME in it, and I'm going to add it to a different chamber in our dish. I'm going to put the cap back on our incubator and start the video.
Alright, well we took a video when we added our no drug and our drug. So, let's look at our results and see what happened. As a reminder, on the left we have no drug, and then on the right we have where we added our drug. So these are two separate compartments of our plate. Let's see what happened. Wow. Alright, so we saw some initial observations. When we added the drug, we had brighter green nuclei compared to the cells that had no drug added. Let's look at a cell in the drug condition a little bit more closely. So we're zooming in on this cell. As you can see at the beginning, we have these green dots. So those are endolysosomes with the dye in them. When we press play, we see that the nucleus gets brighter and the dot gets smaller. So as the dye leaked out due to damage, the dye is going to leak out due to damage, the nucleus is going to get brighter. Let's go back to the drawing board and let's make some conclusions on our experiment. Alright, we're back to the drawing board and we're going to focus on the fourth part of the scientific method, our results. So as we just discussed in the microscope room, the nuclei got brighter, they fluoresced green when we added the LLOME drug. That means that our hypothesis was proven correct. When we added our LLOME drug, we saw that the green dots, the dye that was in the endolysosomes, decreased. So the dye leaked out of the endolysosomes when we added the drug that caused damage. And furthermore, that dye traveled to the nuclei, and the nuclei got brighter. Thanks for coming along on this scientific journey with me today. I had a lot of fun doing this experiment with you and figuring out how that drug, LLOME, causes damage to the cell. Remember, there's a lot more drugs and toxins out there that lead to human diseases that we've got to figure out. And we need eager and excited students like you to join this field. I hope you have a great rest of the day and come back for more. Thank you.